final size of the root. W0 equals initial size of the root at the beginning of the period. R equals relative growth rate. T equals time of growth. And E equals base of natural logarithms. The growth rate obtained here is called the relative growth rate. It is also called efficiency index, as it measures the capacity of a plant part to produce new cells. Thus, we can see geometric growth during the development of the zygote to embryo and arithmetic growth in the later stages of development of the seed into a mature plant. Growth in plants can be quantitatively compared using absolute growth rate and relative growth rate. Absolute growth rate refers to the total growth of a plant part per unit time, while relative growth rate refers to the growth of a plant part per unit initial parameter. Let's compare the growth rate of 2 leaves. Here we can observe a 2 centimeter increase in the surface area of 2 leaves. This shows that the absolute growth rate, or AGR, is same for both the leaves. But the relative growth rate of the first leaf is greater compared to the second one. The difference in the relative growth rate may be due to the difference in growth conditions. Plants require specific conditions for growth. These include water, oxygen, nutrients, temperature, light, and gravity. The increase in cell size is due to the absorption of water. Water also acts as a medium for metabolic reactions required for growth. Oxygen is required by plants for the release of energy in food. Macro and micronutrients are used as energy sources and in the synthesis of cellular contents like protoplasm. In addition, plants need an optimum temperature for proper growth. The growth of plants depends on light, as they manufacture food by photosynthesis. Gravity determines the direction of shoot and root growth in plants. Plant development includes all stages of plant life, from germination of the seed to aging or senescence. In plants, development includes both an increase in the number of cells as well as cell differentiation. We know that root merostem, cambium, and shoot merostem add new cells. These new cells undergo structural changes for specific functions. This phenomenon is known as differentiation. For example, some of the meristematic cells of the procambium lose protoplasm and develop strong, elastic secondary cell walls to form vascular tissues. These tissues then help in the transportation of water and food in plants. In addition to vascular tissues, the procambium also differentiates into parenchymatous cells, which act as supporting tissues. Under certain conditions, some plant cells have the ability to regain the capacity of division after differentiation. This phenomenon is called de-differentiation. Differentiated parenchymatous cells de-differentiate to form meristematic cells. that is, the interfascicular cambium. Meristems formed by dedifferentiation give rise to cells that lose the ability to divide but mature to perform specific functions, which is called redifferentiation. For example, the meristematic cells of the interfascicular cambium redifferentiate into the cortex. Differentiation in plants is open similar to growth, because cells formed from the same meristem differentiate into different structures at maturity. The final structure of a cell at maturity is determined by its position in the plant. For example, cells distal to root apical meristems differentiate into root cap cells, and cells at the periphery differentiate into the epidermis. Plants also develop different structures in response to different phases of life or the environment. This phenomenon is called plasticity. Heterophyly, the presence of more than one type of leaf, in the coriander plant is an example of plasticity in response to different phases of life. As you can see, the leaves of juvenile and mature coriander plants differ in shape. In the same way, the different shapes of the buttercup leaf above and below water are examples of plasticity in response to different environments. In the life cycle of plants, cell growth, cell differentiation, and cell development are interrelated and controlled by intrinsic and extrinsic factors. Intrinsic factors can be intracellular, such as genes, or intercellular, such as growth-regulating chemicals. Extrinsic factors include light, temperature, and the availability of water, oxygen, and nutrition. In plants, growth regulators or PGRs intrinsically control development. These regulators are chemical compounds and are also called plant hormones or phytochromes. PGRs include auxin, gibberellin, cytokinin, abscisic acid, and ethylene. They are divided into two groups: plant growth promoters and plant growth inhibitors. Plant growth promoters enhance growth activities like cell division, cell enlargement, flowering, fruiting, and seed formation. Auxin, gibberellin, and cytokinin act as plant growth promoters. Plant growth inhibitors respond to wounds and stress from biotic and/or abiotic factors by inhibiting growth. Abscission, the falling of leaves due to scars, and dormancy, or the quiet period of the plant during winter, are due to the action of plant growth inhibitors. Abscisic acid acts as a plant growth inhibitor, while ethylene can act both as a growth promoter and inhibitor. The discovery of PGRs was accidental. The discovery of auxin was initiated when Charles Darwin and his son Francis Darwin observed the coleoptile of canary grass exhibiting phototropism towards blue light. After several experiments, they found that the tip of the coleoptile is responsible for the bending of the full coleoptile. Later, F.W. Wendt discovered that a substance at the tip of the plant was responsible for its growth and called it auxin, which means to grow in Greek. But the chemical structure of